Our next topic for today will be the meristems. So again, at the end of this lesson, hopefully you'll be able to identify the different plant cell types, describe different plant tissues, differentiate meristematic and non-meristematic tissues, and explain how different plant tissues aid in growth and development of the plant. Okay, so consider the plight of a green plant. Rooted in place, it seems vulnerable and defenseless against drought, flood, and wind, fire, and hungry herbivores. Yet plants dominate nearly every habitat on land. Now the question is, how do they do it? In many ways, plants owe their success to modular growth. So to understand modular growth, imagine a landowner who plans to build a motel. Money is tight at first, so she starts with just a few rooms. As her business grows, however, she adds more units to the basic plan. Plant growth is similar. Shoots become larger by adding units or modules, consisting of repeated nodes and internodes. Roots can branch repeatedly as they explore the soil. Some plants, such as dandelions, have determinate growth, meaning that they stop growing after they reach their mature size. Most plants, however, can grow indefinitely by adding module after module. Think of it, think of like ivy climbing a building. Such indeterminate growth can continue as long as environmental conditions allow it. So example for indeterminate growth would be a majority of tomato varieties, while most bush-type plants follow determinate growth. All of plants' new cells come from meristems, which are regions that undergo active mitotic cell division. Meristems are patches of immortality that allow a plant to grow, replace damaged parts, and respond to environmental changes. Most plants have two main types of meristems. So we have the apical meristems, which are small patches of actively dividing cells near the tips of roots and shoots. When cells in the apical meristem divide, they give rise to new cells that differentiate into all of the tissue types described in the previous section. Woody plants also have lateral meristems, which produce cells that thickens a stem or a root. A lateral meristem is usually an internal cylinder of cells extending along most of the length of the plant. When the cells divide, they typically produce tissues to both the inside and outside of the meristem. So first we have apical meristem. Primary growth lengthens the shoot or root tip by adding cells produced by the apical meristem. So the picture that you see here is an example of an apical meristem, specifically a shoot apical meristem. Okay, so new cells originate at the apical meristem. The daughter cells eventually give rise to ground tissue, the epidermis, and vascular tissue. The stem elongates as the vacuoles of the new cells absorb water, pushing the apical meristem upward. Meanwhile, new leaves originate on the flanks of the meristem. Okay, so remnants of the apical meristem remains in the axillary bud that form at stem nodes. These buds may either remain dormant or awaken to form side branches. When a shoot loses its terminal bud, cells in one or more dormant axillary buds begin to divide. The result is a bushy growth form. Gardeners exploit this phenomenon by pinching off the tips of young tomato or basil stems, a practice that promotes the growth of side branches and therefore greatly increases the yield. So the picture that you see here are the axillary buds that I am referring to. Next, we have the root apical meristem. So, roots also grow at their tips. Just behind the tip of each actively growing root is an apical meristem, as you can see in the picture. Some of the cells produced at this meristem differentiate into the root cap. Some of our other cells elongate by absorbing water into their vacuoles. As the cells become larger, the root grows farther into the soil. Beyond this zone of elongation is a zone of maturation, in which cells complete their differentiation and mature into the functional ground, dermal and vascular tissues that make up the root. So this is the area of maturation that I was talking about, and this is the area of elongation. This part here is the area of cell division. This is how a root apical meristem would look like under the microscope. So again, this part here is the zone of elongation. This is the root cap. Next, we have lateral meristems. So in many habitats, plants compete for sunlight. The tallest plant reach the most light. So selection for height has been a powerful force in the evolutionary history of plants. But primary tissue is not strong enough to support a very tall plant. The increasing competition for light therefore selected for additional support in the form of secondary growth, which increases the girth of stems and roots in woody plants. Okay, so wood and bark are tissues that arise from secondary growth originating at two types of lateral meristems, which are the vascular cambium and cork cambium. These meristems occur in gymnosperms and many eudicots, but not in monocots. So we have vascular cambium. So vascular cambium is an internal cylinder of meristem tissue that produces most of the diameter of a woody root or stem. This lateral meristem forms a thin layer between the primary xylem and phloem. When a cell in the vascular cambium divides, it produces two daughter cells. Okay, so it produces two daughter cells. One of the two cells remains as a meristem cell. If the other cell matures on the inside of the cambium, it becomes a secondary xylem. If it matures to the outside, it becomes a secondary phloem. And this is how a secondary phloem and a secondary xylem would look like. So this picture is actually going to the center, center of the plant. So this is center going outwards. Okay, so a stem or roots would consist of a secondary xylem, and it can accumulate to massive proportions. For example, a giant sequoia tree in California is 100 meters tall and more than 7 meters in diameter. Secondary phloem occupies much less volume. This tissue forms the live innermost layer of a bark, a collective term for all tissues to the outside of the vascular cambium. So this is the bark. Next, we have the cork cambium. So the cork cambium is a lateral meristem that gives rise to parenchyma to the inside and cork to the outside. Cork consists of layers of densely packed waxy cells on the surfaces of mature stems and roots. The cells are dead at maturity and form waterproof, insulating layers that protect the plant. The cork used in wine bottles come from oak trees that grow in the Mediterranean region. Every 10 years, harvesters remove much of the cork cambium and the thick cork layer, which grows back. Cork is also important in the history of biology because, again, remember, in 1665, Robert Hooke became the first person to see cells when he used a primitive microscope to gaze at cork. Okay, so wood is durable and useful. That's a fact. Few plant products are as versatile or as economically important as wood. Lumber forms the internal frame that supports many buildings. Firewood provides heat and cooking fuel. Most paper comes from wood. Throughout history, humans have fashioned wood into furniture, pencils, cabinets, boats, baseball bats, serving bowls, roofing shingles, jewelry, picture frames, and countless other items. The internal anatomy of a tree trunk is seen in this picture, including the bark, the vascular cambium, and secondary xylem. Nearly all of the trunk consists of secondary xylem or wood. So this cross-section reveals that the heartwood or innermost wood is darker than the sapwood or the outer portion. This color difference arises as the tree ages. The lighter colored sapwood, located nearest the vascular cambium, transports water and dissolved minerals. Meanwhile, as the years pass, the oldest secondary xylem at the center of the trunk, which is the heartwood, gradually becomes unable to conduct water. As its function declines, dark-colored chemicals accumulate in the heartwood. So that's the reason why you'll see that layers of a tree differs in color. Another feature of a trunk's cross-section is tree rings. In temperate climates, cells in the vascular cambium are dormant in winter, but they divide to produce food during the spring and summer. During the moist days of spring, the vascular cambium produces large water-conducting cells. During the drier days of summer, new wood has smaller cells. The contrast between the summer wood of one year and the spring wood of the next highlights each annual tree rings. Fortunately, it is not necessary to cut down a tree to see its growth rings. A slender core extracted from the trunk reveals the pattern without harming the tree. By counting the rings, a forester can estimate the tree's age. Growth rings also provide clues about climate and significant events throughout a tree's life. A thick ring indicates plentiful rainfall and good growing conditions. Narrow tree rings may reflect stress from herbivory, disease, or fierce competition for light or water. A fire leaves behind a charred burnt scar, as you can see here in the picture.